Thanks, Frank and Nancy, for putting together this session. It's really great to be here. And I'm excited to actually tell you a little bit about some of the progress that we've made for monoclonal antibody discovery here at Lampire. The focus of my talk will really be on two formats of antibodies, VHH nanobodies, and also bovine picobodies, given their potential use in bispecific and multispecific antibodies. But really, this platform is fit for purpose platform and can be utilized across all different types of animal species for monoclonal discovery. And I'm actually not. Is this what yeah. Oh, great. Yeah. All right. So just one slide, brief introduction to Lampire. You may not know us. You may know us. We have been around for a long time. We work with a lot of pharma, biotech, as well as academic groups. We provide a whole host of different blood-based sort of reagents, as well as custom monoclonal and polyclonal antibody discovery work. We've been doing this for decades now. It's primarily located in Pennsylvania. We have several farm facilities. We have more than 7,000 animals, both large and small animals. It's quite impressive. We're happy to host your visit when you would like to come and visit us. It's really quite beautiful facility that we have. In particular, there's a facility in Everett, Pennsylvania, that rolling hills of Pennsylvania. It's where more than 600 acres, and a lot of our larger animals roam the hills and the pastures. This again includes a lot of different types of large and small animals. Donkeys and goats and sheep and llamas and alpacas, which is I'm going to talk about, as well as smaller animals like chickens and rats and mice. The one thing that I want to put across there early is there might be a little bit of an underappreciated understanding in just diversity or the level of complexity in the immune systems of animals. We all know T cell and B cell biology to an extent and how an antibody is produced. The immune system is engaged and, B cells and T cells do their thing. But in reality, each species, when it gets down to it, is unique. There's gene conversion in rabbits. There's IgYs in chickens. There's single domain antibodies in camelids. There's picobodies, these ultra long CDR3s in bovine. Each species has a unique aspect of their immune system. And again, animals are really much our friends. We're highly accredited. We're ALAC accredited. We are ISO certified, and we meet or exceed as needed any sort of like EU or UK based animal welfare care of use standards. All right. So, and as an introduction to VHH nanobodies, which you may be aware of, I thought to share this slide, and what this really is kind of a brief history of VHH as therapeutics, and it kind of goes from left to right. On the left side, you're maybe familiar with caplacizumab. We actually saw a talk just yesterday from the Sanofi folks of caplacizumab. This is the first VHH-based therapeutic approved. It's on the market. And it really is a fairly simple design. It's a bivalent molecule with a little linker in between. Very short half-life, right? Doesn't last long as by design in the patient. Going to the right, you'll see some next generation VHH-based therapeutics, like the one that is being developed by Boehringer Ingelheim. This is actually a trivalent and a tetraspecific antibody that is, targets VEGF and Ang2, but it also has the albumin binding domain. And what is that for? That's for a half-life extension, right? So that's a relatively small VHH domain that, provides half-life extension and improvements in PK. The molecule next to that on the right is an example by Inhibrx. And this is an extreme example. And there's several elements of this. It's kind of cool. It's a hexavalent molecule, so it has 6 binding domains against OX40. And this is a type of wild sort of molecules that you can envision. It's not wild just for the purposes of being wild, but it really, when you do this sort of thing, you're really leverage avidity, and potentially, when you develop these sorts of molecules, you really use that avidity in your favor to drive cis or trans sort of receptor interactions and really drive efficacy in vivo. This one also has an FC domain. I can't remember if this one actually was FC engineered, but the FC domain, of course, is also your classical FC IgG1 base that extends half-life. Some other examples, I'm not going to go too far in, but we've also put these into ADCs. The one example is a CTLA-4. It's a molecule that I was proud to be involved with during my time at Molecular Templates. It's a biparatopic used in tumor microenvironment to get basically kill T regulatory cells. And Carvykti, a CAR T based BCMA, biparatopic again. What I don't know, and maybe someone could tell me, is, and I'm assuming that there's some, bispecific VHH ADCs in the clinic. Probably there are. I don't know. Tell me if you know of any. And of course, we know there's a lot of promise with in vivo CAR T and VHH molecules. So again, just to kind of highlight the diversity of VHHs. Now, when I said the title of my talk, sequence forward approach. What is a sequence forward based approach? Well, I've kind of described it here, and I'm not going to capture all the subtle aspects of it, but really it starts with deep sequencing of the immune repertoire. So we engage an animal, we immunize it, and we specifically focus in our platform on identifying and sequencing quickly the antigen-positive memory B cell population, and we get thousands of sequences, thousands of VHHs here. As long as there's a robust immune response, we'll get thousands of VHHs. We get the full-length sequences. So it's immune repertoire, but we're not just looking at CDR3. When we do our studies, we're looking at the entire V gene region. So from framework 1 to framework 4, CDR1, 2, and 3. We need to look at that. When we work with our clients, you get all the sequences, a curated list, run through our sort of analysis. You could run it through your analysis. We could deliver you raw sequence data, whatever you like. We're very flexible, and importantly, IP and royalty free, right? So we make no claims whatsoever on any of the sequences that we would deliver. And then the in silico triage. So this is an important aspect of this and a key part, right? So if you have this subset of sequences that you pull out from the animals, and this really represents the immune system's response to a given antigen, there's going to be a lot of diversity there, right? And so how do you triage those sequences? Well, of course, you can triage them based on many different things, right? Not all of them are fit for purpose for every type of molecule that you're designing. But certainly looking at sequence diversity, CDR3 clonotype, there's actually sequence signatures that we've learned over the years that are actually indicative of molecules that have gone through, affinity maturation events beyond just your somatic hypermutations. But there's other sort of, sequence-based characteristics that we were aware of now. You could also apply humanness scores and kind of a path to humanization. Of course, VHHs are generally IGHV3, so that's pretty much a commodity, right? So to humanize a VHH coming from an llama alpaca, you know what? It's not that hard. Really, it's not that hard if and when you need to do that, because the truth is that you don't always need to do that. A good percentage of VHHs directly from the animal actually essentially are at or above the humanist levels found in clinical phase molecules. So again, just to highlight the fact that a lot of these molecules are coming out, kind of ready to go as far as a developability sort of point of view. But then you also need, of course, want to look at developability. You could look at a sequence level, remove those early liabilities that are going to come up and be problematic in the future. And you leverage AI/ML, right? And I'm not going to talk about AI/ML. I think I might have one slide in this talk that I used AI to generate. You'll probably know it when you see it. But certainly, using these sort of algorithms to understand your sequence, in relationship to paratope, epitope, and ultimately function, is something that can be done nowadays, and really kind of, something that we could continue to leverage in such an approach. And then point number 3, and this is something that I'm going to a message that I want to drive home a little bit, and I have a slide on this, and the concept of in vivo maturation. We're not always able to do this to fit within the timelines of a given project, but it is something worth considering that's unique about large animals. And the fact that when you collect VHHs from large animals, it's not an endpoint. It's it's a blood draw, just like as if you're going to give blood at Red Cross. That animal, the blood is drawn, and then the PBMCs are taken in VHH, and you do your discovery. But the animal's not going anywhere. If you want to re-engage that animal, you can. So, what is the platform? Well, here's a real kind of a busy summary slide, and I'll walk you through it a little bit. There are some key aspects of it. It really starts with the llama immunization, right? And essentially, we've done probably thousands of different llama immunizations at this point. The one key thing here is that nowadays, if you're doing in vivo antibody discovery, think about what you can do with mRNAs and saRNAs. Really, it works great. It really does. Of course, you could still certainly do a campaign with your standard proteins and cells and DNA, but we've really seen and we've optimized protocols with mRNAs and siRNAs. And you can imagine if you're a protein or antibody person, the types of unique, let's say, target molecules that you can make through an mRNA, saRNA works great. Consider it if you're doing these sorts of studies. After the immunization, collect the PBMCs. This could be from one or more animal and one or more time point. And I'm going to get to this point because this one or more time point is a part of that in vivo affinity maturation event in order to leverage that. You take those PBMCs, now you put them through, in our platform, it's a flow cytometry or a FACS-based platform. Of course, there's a lot of different ways to do this, and we can work with you if you're doing phage display, if you're doing yeast display, or if you're doing 10x genomics or beacon systems, and they're all good systems. Each system has its advantage and disadvantages. What we've developed is we worked on what was a gap for single B cell discovery. And I've been doing single B cell discovery for more than a decade now. And there was always a gap in being able to do efficient single B cell discovery from these, something like a llama or a pack or some of these other off-used, less used animals. The gap has been the phenotypic markers. So we've invested in the ability to target the antigen-positive memory B cell population that we want to target. And that's the key driver of this technology, is building out that FACS-based isolation of those cells. As part of that FACS, we use all different kind of baits, right? So it's got to be some form of your protein, even if it's like a GPCR or an ion channel. It needs to be some, a VLP or a nanodisc or even cells. We could take your cells and we can make a membrane preparation and use that as a bait to get those antigen positive cells out. And then once we go through the FACS, we go right into the sequencing. So that's the sequence forward approach. We get those sequences early. I'm not going to go through that again, and that goes forward into recombinant mAb production and screening. We also have the ability to do your secondary screening or more complex sort of bispecific and multi-specific formats. We have our favorite bispecific format for VHHs. Your kind of tandem VHH that always really expresses quite well. That may not be your end format ultimately, but we do have the ability to do that. We also have the ability to do epitope affinity, as well as more advanced sort of cell screening and functional assays. So that's the platform. So this idea of in vivo affinity maturation, so in this is a real world example in which we started an immunization on the left in a cohort of animals. I think it was 2 llamas with a full length human protein. We took the PBMCs. We isolated a good amount of diverse binders. This was a target. We had to find novel epitopes, and specifically this was being developed for a mechanism of action that really benefited from having a biparatopic design, and particularly one against a proximal domain and one against a distal domain. But it was really that proximal domain, the affinity of that proximal domain binder, which was really driving efficacy in this platform. And we found a 40 nanomolar, and that's the one listed there on top, that 40 nanomolar clone during that first round of immunizations. And again, when we generated these molecules, and we were also doing phage display, and we're generating some nice molecules, and we're learning along the way. But again, we learned that we needed to get a higher affinity engager at that proximal domain. So one of the things that we did is shown on the right is essentially we re-engaged that same animal with a chimeric antigen. I'm not going to go into the concept of it, but it's a unique way to, let's say, target specific domains. And in this regard, we targeted the proximal domain. What we found is we found clones, and again, this is just a very shortened list, right, for the example here. But you could see after we did this rest and rededicate, or did this in vivo affinity maturation, re-engage the same, we found binders within the same CDR3 clonotype that were a lot higher affinity. And these actually went on to be the lead of this project ultimately. So an example of in vivo affinity maturation, something that you could do in the large animals, rather than some of the endpoint sort of studies that you might be doing in some of your smaller animals. Okay, so second part of my talk is something that I haven't really, disclosed some of this information. It's something that we're really excited about at Lampire. And I don't know if you know who this cow is, but it's actually kind of a famous cow. She lives in Austria. If anybody knows, you could yell it out. You probably don't know. Do you know? No. Her name is Veronica. And what she's known for is her tool use. She's learned how to use tools. So she's a very smart cow. And she's used these tools to kind of scratch her back, But what I'll tell you is, cows are smart. Their immune systems are smart. And what we've learned in recent years is that cows, bovines, actually make a unique type of antibody. And it's really a conventional antibody. It's a heavy chain, light chain. In this case, it's a lambda light chain. But they make what's called picobodies. You've probably heard of these. Maybe you've seen them described as ultra-long CDR3s or even knob domains. But essentially, it's what we call picobodies. And what these are, they're exceptionally small. So kind of almost like a nanobody, like a VHH nanobody, right? But even smaller, so 30 to 60 kilodaltons. And they're kind of in what I'm going to call a subdomain. So they're really, and you could see in that kind of in the image there, where I've kind of circled the bovine IgG. That is the picobody, or the ultra-long CDR3 that's coming out of the fab, right? That's modular. That has the entire paratope. So the entire binding region is built within that picobody, or that ultra-long CDR3. It's quite wild when you think about it. So that particular type of bovine picobody containing heavy chain antibody has basically abandoned all of the other HCDR1, 2, all the light chain CDRs, and built the entire paratope into this knob and stalk structure in order to really generate a lot of novelty in the epitope space that it could kind of encounter. Very unique thing. I'm a antibody, sequence person, engineer by design, by training. And so, I'm going to show some sequences, but I don't want to bore you too much. But some unique, on the left side, you could see the unique architecture. I'm again kind of looking at the FV region there of a bovine antibody. You're seeing the stalk, right, which is this kind of extending stalk that kind of sticks the knob out in space in order to interact, and that's the paratope, the knob. But this stalk, I will say, is actually quite important. That stalk is generated, and actually, if you look as far as from the germlines, the initial part of that stalk is a germline sequence, but then it gets filled in with the IGHV D region from bovine. But the way that the bovine makes these antibodies is very germline restricted. So there's really just one IGHV, two allelic versions, but just one. And you could see that on the right-hand top there. And the distinguished characteristic, I mean, again, this is germline sequence. We're looking at germline sequence, right? Is that right at the end there, that C cysteine TTVHQ, right? That's the canonical. If you see that, you probably have a pico body, as long as it also has the rest of the pico body place domain. But and then there's just one D segment, which is shown there. The D segment is exceptionally long. And oh my gosh, when you look at the diversity in the animals of what that D segment goes through the inversions and the affinity in the somatic hypermutations, it does not look anything like germline. You know, it goes through an amazing amount of AID and somatic hypermutations. It's quite wild, and there is a specific J segment which does again basically has some of the descending stalk. So there is this ascending knob domain, which is your paratope. There's a wild amount of affinity maturation and somatic hypermutation going on, and then you have your descending stalk. I'm not going to talk too much, but again, the point being here is that although this ascending stalk, at least part of the ascending stalk, is germline as far as being part of the IGHV region, it's heavily mutated in the animal. So you can imagine that D segment, the part that makes the paratope, is heavily mutated and goes through somatic hypermutation. But the stalk regions also are, and this is something that's underappreciated in the field right now. However, one of the things that we're trying to do is we were trying to develop specific tool antibodies against the stalk region in order to try to identify antibodies that would be good use for some of our downstream work, including doing some of the serology, like being able to, let's say, measure the picobody specific response when we're doing, when we're engaging the immune system in these cow cohorts with an antigen. And we found an antibody that's somewhat interesting. You could kind of see it described here. Again, it was a peptide-derived peptide, not my favorite sort of immunogen, but this is what we were working with at the time. We put it through this was actually done in llamas, so we're trying to do an anti-bovine picobody antibody in llama VHHs, and we found some VHHs. And actually, as you can see, when we put it through the bottom left there, you can see we're doing some FACS-based sorting. In that B population is really a picobody-containing memory B cell population that we sorted out. Now, what we found is that it can work. It definitely enriched those CTT-VHQ, so those specific sort of germline-based ascending stalk picobody-containing antibodies, but it was like, in a sense, too specific for our purposes. So it did not capture a lot of the other picobodies that have the ascending stalk. So for what it's worth, we're making this antibody available for anybody who might be interested in using this antibody. We're making it available basically at cost. I can imagine it could be useful for other purposes, and happy to chat with you if you're at all interested in receiving this antibody. Again, and I didn't really touch on it too much, but this ascending and descending stalk be a region that you might think is kind of static, but it's not. In a survey of all the GenBank sequences out there, we've found in our animals, and we've had looked at tens of thousands of unique picobody sequences at this point in time. A lot of them are not really represented in the GenBank databases right now. This is what I'm showing here is an example of what I'm calling some non-canonical stalk-containing picobody sequences. So these are just purely based on the ascending stalk sequences that have no representatives in GenBank, and they were kind of like one-offs in this particular study, one out of, I think, like 5,000 or something. And so the question is, and we did some homology modeling of these novel picobodies with their stalks. We asked the question, is this really functional? Can you even express it? And the truth is, yeah, you can. It's highly expressible. So it expresses great. it's, again, the non-canonical ones are the ones that I have highlighted in this slide in yellow, express just fine. So we don't know if these are functional yet, to be honest with you, but it's something to be mindful if, just for, if you're ever start looking at bovine picobodies, is that I think it's important to understand that, the field is still growing out there. Our understanding of the diversity, of the true diversity of these picobodies is still growing. There's a lot of great literature out there, but I don't think we really know the true diversity just yet in these picobodies. And, and of course, since I'm, talking at this section on multispecifics and picobodies, I had to show this slide. And this was some work that we started to do in formatting these things into different bispecific and multispecific formats. And it was really eye-opening. So what I can say is that they play well. This work that I'm showing here was minimal amount of engineering, really no engineering, to be honest with you, of the stalk and picobody domain. And we're formatting these into different sort of Fab-based MAB molecules. The one on the left is my favorite right now. It expresses quite well. What you're really doing is you're putting the picobody in either the heavy chain of the CDR3 heavy chain, or the CDR3 of the light chain. And so you're potentially making a bispecific, very easily potentially making a bispecific tetravalent molecule with picobodies in your sort of human or chimeric backbone of choice. This works in several different chimeric human and mouse backbones. And also can work, but I think it's going to require a little more engineering in some of these other formats, and we've had some success. We've had some failures, particularly putting the picobody in like CDR1, CDR2, and that sort of thing. But it's kind of exciting to me because I have worked on a lot of different formatting of multi-specific and bispecific formats, and just to see this sort of modular format and being able to open up some of the potentials out there in using these sorts of picobodies in not only a Fab or MAB-based multi-specific and bispecific formats, but also in other ones as well. So I know I'm running low on time. I'm not going to talk too much. We do have now available a in vivo-based approach where we can immunize cows, just like with llamas and alpacas and other species. They accept mRNA LNPs. They accept self-amplifying, self-replicating RNA LNPs. They work quite well. And we go through a similar process. And we're building out this platform right now for picobody discovery. And this was one AI slide. I'm not going to go through it, but essentially just what are the potential applications of picobodies? Obviously touched on the new novel IP that could be there, the new novel epitopes that you might be able to discover, and a lot of the different formatting possibilities that it was probably just like VHHs that are approachable here. Summary and use case. I won't repeat myself, but essentially, it's similar to like your other species. Doing this sort of work is approachable with the new genetic-based immunogens. It works well for something like GP-SARs and ion channels, as well as more standard sort of soluble proteins. The other point I want to get across is, again, the full diversity I don't think is known just yet. I think this is still building. I've highlighted some of this in the stalk, but we're seeing a lot of novel structures related to the disulfide bondings in these molecules. And again, the novelty of it, being able to afford, if you needed molecules that were, unique epitopes, unique IP, this could be something worth considering. And just to highlight here, again, I think this was a homology model of one of the non-canonical sort of stalks in combination with its picobody that we were looking at. Suffice it to say, what we've learned is that when we go after these molecules, we recover the entire knob and stalk domain because we think that there's some importance there. And with that, I will end. And thank you for your time.
Thank you, John. Interesting platform. I think we have time for one quick question if anybody has one. Yeah, go ahead.
Thank you. Very interesting talk. If I understand correctly, the bispecific construction you did was grafting the bigger body onto the antibody framework. Have you tried to fuse it to other proteins to take advantage of the small size?
I have not yet. We have minimal amount of, so far we've done more like primary screening, kind of like lead ID, molecules. And so, you graft it onto something like a human or a mouse, FC domain, and you could put it right through your standard sort of expression, platforms. But yeah, it's a good question, but we haven't really done that yet. All right, thank you all. I think that's it.